Nucleic acids are made of nucleotides, which are the monomers. Remember that mono means one, so the nucleic acid would be the polymer, many monomers. Okay, that's going to make sense later. So the first and most important component of a nucleotide is the pentose sugar. Okay, for DNA it's going to be deoxyribose. Remember the D for DNA, and for RNA it's going to be ribose. So we have that the pentagon, the sugar is going to have an oxygen on top always. It's going to have 5 carbons. One is going to be outside. We're going to see that later. And it's going to have a hydrogen, or it could have an OH hydroxyl, depending on which one of these two it is. We're going to look at that later as well. So it's always going to have a hydroxyl here attached to the 3rd carbon. And the 5th carbon is going to be attached to the phosphate group, very important as well. Then we have the nitrogenous base, which can be either of these 5. We know that a nucleotide is a monomer, but a nucleotide chain hence makes up a polymer, and in this case it's going to be a nucleic acid. This can be both a DNA polymer or an RNA polymer. Looking at an example, we have that they are bonded by a polymerization, which is a type of condensation reaction, which is when you release one molecule of water and use energy in the form of ATP. So we have that this covalent bond is between the hydroxyl Looking at DNA, we'll see that it has a double strand. So one of them will have the 5th end on this side and the 3rd end on this side, and the other one will have the opposite orientation. This essentially means that it is anti-parallel, and this gives it the double helix shape. Okay, then we have the hydrogen bonds that happen between the nitrogenous bases. So as you can see here, thymine pairs with adenosine, which has 2 rings in its structure. It's a purine. Thymine is a pyrimidine, as we saw before. So they form 2 hydrogen bonds. Cytosine and guanine form 3 hydrogen bonds. And we see that this is based on complementary base pairing, which is very important. We'll look at that as well later. So then we have the covalent bonds that we saw before between the hydroxyl of the 3rd carbon of a pentose sugar and the phosphate of the 5th carbon of a different pentose sugar. Okay, then we have the pentose sugar, which is actually deoxyribose for DNA, and that would be it. If we compare DNA and RNA and we look at their sugar, we know that deoxyribose is for DNA, and this time the difference is going to be that the 2nd carbon is going to have a hydrogen attached, whereas in RNA the ribose sugar will have a hydroxyl attached to its 2nd carbon instead. Other differences involve the nitrogenous bases, so we know that DNA contains thymine, cytosine, adenosine, guanine, and it is double-stranded, while RNA is single-stranded and contains uracil instead of thymine. So complementary base pairing is the following. There's adenosine with thymine in DNA, and adenosine with uracil in RNA, and cytosine, which always pairs up with guanine. So this ensures that the base sequence during DNA replication is always the same. So if you haven't looked at DNA replication yet, I suggest you do so. I have a video on the screen right now, and also a video on gene expression, because it ensures that the same protein is produced every time a gene is expressed or activated. I also have 3 flashcards in the video's description, as well as past papers, so I recommend you check them out.